This week in Pediatric Oncology, the podcast exploring hot topics and exciting advances in childhood cancer. TWiPO is produced by Solving Kids Cancer, nonprofits located in New York and London dedicated to improving research and supporting families, because every kid deserves to grow up. Subscribe to TWiPO through your favorite podcast platform.
This week in Pediatric Oncology, the podcast about new advances for childhood cancer. Hi, everybody. Welcome to episode number 96, recorded on February 15th, 2022. I'm your host or co-host, Tim Cripe from Nationwide Children's Hospital in Columbus, Ohio. We're affiliated with The Ohio State University. I'm here with my co-host, Brenda Weigel from University of Minnesota. Welcome, Brenda.
Hi, Tim. It's a pleasure to be here.
Today we have a guest with us, Kristopher Bosse from Children's Hospital of Philadelphia. Chris, thanks for being here.
Thanks, Tim.
So just by way of brief introduction for our audience, you got your undergraduate degree from Bowdoin College, summa cum laude, and then you were UPenn ever since, Penn Medical School, Penn Residency in Pediatrics, Penn Fellowship in Pediatric Hematology Oncology. And looking through your CV, you've gotten pretty much all the possible awards and scholar grants and training grants from every national organization I can think of. So congratulations on all your success today. You're amassing quite a career. So we wanted to talk mainly, though, about your work in Glypican-2, GPC2, since it seems like it's really taking off and you've had a lot of success with that, enjoyed reading your prior publications. So could you just start off by giving us a little bit of a background on how that project came to be? What's the history of it? How'd you come up with it?
Sure. Well, thanks for the introduction, and it's great to be here. So the project really started when I was a postdoc after my clinical hematology oncology training, and joined the lab of John Maris, who's a pediatric oncologist at CHOP, and was really tasked with a project to discover new immunotherapeutic targets in this disease, high-risk neuroblastoma. And it was right around the time where, pediatric immuno-oncology was really taking off, and John and Crystal Makel had formed a team where they sort of combined their expertise of genomics and immunotherapy to really get after this problem of developing new immunotherapies for several pediatric cancers, and our cancer was neuroblastoma. So at the time, this was a new thing, and now a lot of people have done similar studies, but we use RNA sequencing data to just compare a large set of neuroblastomas versus normal tissues, and just ask the very simple question of what's expressed in neuroblastomas, what isn't expressed in most normal tissues. And I think really realizing that we didn't really know what that differential expression needed to be. So it's never perfect, of course. So we got a list of interesting genes, and then I spent better part of the next few years really validating these at the protein level, and then exploring the functionality of them, and then developing binders collaboratively across our team, targeting them. And GPC2 just happened to fall out of that analysis. It was a reagent, or it was a protein where we actually could find reagents that worked, which is part of the challenge, because in any of these genes, there's very little known about them. And it continued to look good, throughout our pipeline that we were developing, and we haven't left ever since. So it's been a lot of fun, and we're very close to, I think, at the back end of this, getting GPC2 directed therapies to the clinic. So it's just been a whirlwind of fun, and I've learned a lot about immunotherapy, and I think we've learned a lot about how to what makes good targets, and I think there's several other targets coming through that have built upon the pipeline we developed.
And Chris, building on that, I think many in the audience are familiar with in neuroblastoma targeting GD2 with drugs like dinutuximab that have become part of kind of standard therapy right now for high-risk neuroblastoma. What makes GPC2 a better or different target? And can you sort of compare and contrast between what folks know in targeting GD2 and why this might be beneficial to look for other targets?
Yeah, I think I just first say, I think we need many more targets. I think in the day where we're learning that antigen loss, whether it's GD2 or GPC2 or CD19, is becoming the major mechanism of relapse. I think dual antigen targeting and it gets at the project we'll talk about, but is really important. And so more antigens, the better, I think. And in disease like neuroblastoma, GD2 been known for decades to be high on several tumors. But it wasn't discovered in an unbiased fashion. It was discovered by, I think, antibodies are floating around in the blood of patients with melanoma and neuroblastoma. So, I think for those of us who treat kids with neuroblastoma with dinutuximab, it's obviously revolutionized how we treat these kids, both in maintenance therapy and first salvage at the time of relapse. But these kids require sedation, infusions. Some kids continue to have progressive disease despite GD2 targeting. It's controversial, but how much GD2 is actually in nervous central nervous system, and does that preclude giving CAR T cells or others to brain tumors or CNS neuroblastomas? I think it is unclear, although there's some good data coming out of Crystal's lab that suggests that's not going to be a problem. But I think it's really the morbidity that comes with dinutuximab, the fact that obviously kids relapse with GD despite dinutuximab, and I think more data we get on those relapses, it is showing GD2 downregulation. So I don't think it's so much that GD2 is a terrible target. I think we just need more targets. And I think GPC2 is maybe a bit cleaner from a normal tissue perspective. We'll see about the toxicities when we get into the clinic. I really think we just need to develop additional targets to have things that we can take off the shelf when we can sequence these. So when we do get antigen loss, we'll have other backup therapies, or we can do bicistronic constructs, things like that.
You bring up a really good point about the normal expression, because I was looking through the human protein atlas, which anyone that's listening can look up, comment, publicly available website. You can see where the expression of any gene or protein is, and it looked very absent from most normal tissues. So there's a lot of criteria that that a target has to fulfill: be high enough expression on the tumor, selective enough, not on normal tissues. How many other targets do you think it's possible, or have you encountered in this project or subsequent projects? Do you think we're going to be able to have a lot of targets? There's a lot to ask of a target, I guess, is what I'm saying. And how lucky can we get?
Yeah, I think I saw some quotes somewhere that said there is no perfect solid tumor immunotherapeutic target, or we've been looking for decades and we still haven't found them. And so I think that is true. There isn't perfect. And what I think we just thought was an ideal target a decade ago, I think we've changed how we think about that just because of experiences and looking at some of these. And so there is not going to be a completely negative normal tissue target. And GD2 is not that. And I think we have to develop ways where we can sort of perhaps thread the needle or have a high enough therapeutic index where we can tolerate some normal tissue expression. So that's one thing. How many targets are there? We are developing others in the lab, but it's a tough business. And I think a lot of these fail, later on down the line after you take a deep dive because you learn something, or, you see some normal tissue expression that you didn't expect. Now, how much of that is relevant in the clinic? I don't think we have any idea, right? We don't know how much normal tissue expression is tolerable, what expression matters, how high. It's really hard to compare. If you do see something on the human protein atlas, how that compares to tumors, right? Those aren't done in parallel. A lot of those are adult tissues, or most of those are adult tissues. So I think we're going to learn a lot from the GPC2 experience, and we know what that looks like in our studies, and then how will that perform in humans. And I think we have several other things coming down the pipeline where we'll learn sort of the basic studies we did in the lab and how that correlates to in the clinic. We never had that for GD2, right? GD2 is a sugar, so it's hard to do any of these standard analyses. There's no, you can do GD2 IHC, but it's not very good. There's no RNA to compare. So we don't really have a sense. It's very, very high in tumors, but what that means in all these preclinical studies, we just don't really know. So I think this will be a good target to understand those things and learn, and then hopefully go back, and maybe there are a lot of other targets where we said weren't good because we were afraid of something, and then they may come to light once we learn a little more.
And Chris, you've really highlighted some of the key elements of target selection and the challenges of identifying those and of really sort of figuring out what are the relevant factors, et cetera. But there's also the issue of how do you hit the target, and how do you design something that effectively will kill the cancer cell. And as we said, the expression on normal tissues and et cetera. But can you speak a little bit to sort of how you went down the pathway of deciding an agent that's considered an antibody drug conjugate rather than just a straight antibody? What are some of the challenges that you face in developing targeted strategies using antibodies?
Yeah, I think we don't know which is the best in kids, right? There are CAR T cells surely in kids. There are antibody drug conjugates. Most of these are all in the leukemias. And so I think which therapy is going to be most efficacious for GPC2 and neuroblastoma, we don't know. So we've taken the route of trying to develop everyone we can, and not everyone. So antibodies, we looked very early. We had antibodies, which all these therapies came out of. We never saw any antibody ADCC or any of that activity with any of our antibodies. So that was, sort of precluded an antibody only therapy, much like dinutuximab works. We have close collaborators on our team who are ADC experts, and they decided to make one of these, and we started testing. It looked really potent. So that was, sort of the easy answer to the first route we went. But in parallel, we're making CAR T cells. It just took a little bit longer to get efficacious ones. But now I think we're at the point where we have really potent CAR T cells and really potent ADCs. And I think we've learned a reality in pediatric cancers and in academia where we can actually bring CAR T cells to the clinic largely in academia. We have the processes set up. Surely at CHOP and Penn, we have those processes set up. So that's something we can develop in academia. ADCs are harder because it's harder to get. We don't have those processes here. So we really tried to collaborate with industries on ADCs, and we've had some collaborations that haven't, continued, in large part because I think companies really want to use their favorite payload, and not every payload, it turns out, works in neuroblastoma cells. So we actually published a paper back in the fall just testing a bunch of different payloads in neuroblastoma cells to really get at the question that some payloads are good, some are not good, and an ADC with a payload that isn't as efficacious isn't going to work. So we have a really exciting new collaboration that's building with a new company. So we do hope to get an ADC to the clinic too. But I don't think we know, and I don't think we understand which is going to be most effective for pediatric solid tumors. And if we're able to get both of these, we'll have an ADC and a CAR T cell in the clinic from the same binder or antibody. So I think it will really give us an opportunity, if we can do that, to really understand, with like materials, what is most efficacious. And there's probably a role for both of them, and both, even for GPC2 in the clinic, at least we think so. So we'll have to see.
And Chris, thank you for highlighting those things. And you've used the word solid tumor and taking this work beyond neuroblastoma. Can you speak a little bit to what you know about GPC2 or other GPCs with respect to other pediatric tumors, and how some of that targeting may be different or different strategies might be needed?
Yeah, so it seems, and I don't say something, a little proof for it, but it seems that every tumor has its own glypican. And so there's a lot of redundancy in how glypican-2 and other glypicans are signaling co-receptors. They largely act to facilitate ligand receptor binding at the cell surface of both normal tissues during development largely and in cancer cells. And they drive hedgehog, Wnt, any pathway you can think of important development in cancer. So, but it seems like most tissues have one elevated, or most cancers have a one elevated glypican. For neuroblastoma is GPC2. For hepatocellular carcinoma, which is probably the most well-known, is GPC3. And there's lots of immune therapies targeting GPC3. GPC3 is a high in Wilms tumor, and you can go on down the line. So I think there's redundancy biologically, but GPC3 actually is a good example of, I think, many programs that are targeting this in the clinic and hepatocellular carcinoma. It's a major one. There's also a pediatric program coming out of Texas that are using CAR T cells to target GPC3 in several pediatric cancers. So we know a lot about, and I think part of our so-called pipeline was really this area. What's in neuroblastoma, but what else? What other tumors express high levels of GPC2? So we're lucky, I think, as genomicists to be able to do those analyses pretty easily in the day of RNA sequencing. It's a pretty quick thing to look at, and it turns out that high-grade gliomas have a subset of high GPC2 expression, and we have a paper under review that's showing targeting in high-grade gliomas. Medulloblastomas have very high GPC2. This tumor, I'm really interested in. I think it's like a neuroblastoma-like brain tumor biologically. It's an embryonal tumor of multi-layered rosettes, which is rare, but a very lethal tumor, usually to young kids. Retinoblastomas are very high. They're paired CNS tumor pineoblastomas. And then we developed some data, and this was largely to help get some of these to the clinic in collaboration with the industry around small cell lung cancers. And I don't know much about small cell lung cancers, but I know they're neuroendocrine derived, and they have a very similar surfaceome. So you have some data to show both for ADCs and CAR T cells that GPC2 targeting is efficacious, and it's actually proven to, I think, be an important thing as we try to sell our therapies to companies and try to not sell per se, but sell them speaking and really, try to get people excited about this. We're in a world where all the cancers, thankfully, we treat are rare, but which makes it a little bit, hard to convince companies to invest all this money in helping us develop these. But the lung cancer angle has, I think, helped a little bit. And we always have we won't just go after lung cancer, of course, but if that's part of a GPC2 program, all the better, I think. So, I think we're lucky that RNA has predicted GPC2 expression well, but we've also learned that RNA isn't always good for a predictor for some targets. So, I think there's other ways to profile tumors, of course.
So it sounds like it's very potentially widely applicable, which is very exciting. And I assume the majority, you haven't said like what percentage, but the majority of neuroblastoma, is it tied to risk of neuroblastoma? Is it in the high risk, not the low risk? Is it prognostic? Just wondering a little bit more about the applicability across that.
Yeah, that's a good question. So trying to think of how we figure this out. I'm not even sure how we got to this, but it's actually transcriptionally regulated by MYC-N, which, as you know, is a oncogenic transcription factor in neuroblastomas and other cancers, and it's a marker of aggressive disease in neuroblastoma. So that's we've shown both in neuroblastomas and small cell lung cancers, and it's also encoded on chromosome 7q, which in the world of neuroblastoma segmental chromosome aberrations is not commonly talked about, but it's actually quite frequently gained at a low somatic gain in 50% of neuroblastoma tumors. So we can see both of those regulate GPC2 transcriptionally. And then it ends up being a lot of these tumors that I rattled off a few minutes ago actually are pretty commonly have 7q gain somatically too. So, or MYC-N amplification. The question of MYC, not c-MYC regulation is unclear to me. And we've tried to look at this, and it's not clear whether it's also regulated by c-MYC, but that's a possibility. So we think those are good biomarkers, but I will say that, ends up being a neuroblastoma, MYC-N and 7q gain are basically mutually exclusive. So it does get at there's one or the other, but as you mentioned, that most neuroblastoma, it's not every single neuroblastoma, but it's above 90% express GPC2 for sure.
So you've got a good target, you've got strategies to attack it. Where are you with respect to clinical translation, either launching trials or moving it forward to the next steps?
Well, I spent most of my morning this morning working on a clinical protocol for a GPC2 CAR. So, hopefully that will see the clinic soon. It's been an exciting collaboration with a local company called Community, which is comes out of Carl June's lab. And our excitement to get that is, it's been a bit slow during the pandemic, but I think we're close to getting it launched, and it'll be open at CHOP for patients with neuroblastoma initially. And then we hope to expand it with some companion diagnostic IHC assay to identify some of these other tumors that have GPC2 expression. So we're close. And then hopefully an ADC is not too far to follow. But again, that's been a little more complicated.
Are you also making a bispecific, or putting everything sort of into those two buckets?
Yeah, we have not dove deeply into bispecifics, but that's more of a bandwidth. I don't know. I mean, I think bispecifics are much like ADCs. They're I know Nate Colling's done a lot of this, but they require a platform to really build these up. There's so many different iterations and engineering of the the templates per se that a bit weary to go after, because I think we're not really set up to do that. We've done a little bit of it, but there's no reason to think that they wouldn't be just as efficacious. We just don't know.
It's very exciting to hear that this will be translating into the clinic very soon, Chris. That's super exciting. And as we only have a few minutes left, I wondered if you could give us a hint as to sort of the major directions that you see your work and the work of your lab going in the near future, in addition to the exciting opening of a CAR T trial.
Yeah. We'll go where the science leads us, but I think for now, we're all in on GPC2 targeting, and I think as Tim just mentioned, it's there's a lot we could do. We're learning a lot about immunotherapy, CAR T cell therapies. I don't see us becoming a real CAR T cell therapy, but using the engineering to really get at targeting this disease and staying very translational and getting what we developed to the clinic. We are working on some next generation CAR T cells based on data we have, and we'll see the clinical biological correlative data from the trial. And then we have a few other targets we're interested in, and I do see us developing some bispecific approaches. Again, I'm a genomicist. I think I'm a cancer biologist. So we'll see. I think I do neuroblastoma now. I can see myself branching on into other aggressive cancers. There's the opportunity, to target, bring some of this technology, and as you guys know, we have a lot of very aggressive, lethal cancers in the clinic, and again, thankfully they're all rare, but a lot of them, a lot of us study neuroblastoma, but there's a lot of other rare malignancies that actually have, even less therapy. So, I think there's a lot of things we could do, and especially just in this realm, and target discovery for those cancers too.
I'll ask Tim if he has any final questions or comments.
Only that, it's very exciting. We need more of these targets. I'm glad you guys are making a lot of good progress on it, and congratulations on all your work so far. And thanks for sharing with us today.
Thank you, guys. It's been a pleasure.
Yeah, and I have to say thank you very much to Dr. Chris Bosse for this wonderful overview of targeting GPC2 in neuroblastoma, but other cancers as well, and really the concerted effort to bring those discoveries to the clinic is really just fantastic to see. And congratulations on your tremendous work. And it looks like that's it for this week. And I want to thank again Dr. Chris Bosse, Dr. Tim Cripe, and most of all, thanks to the team at Solving Kids Cancer, a nonprofit charity dedicated to improving survival through creating novel treatment options for children. Remember, the more we learn, communicate, share ideas, and work together, the faster we'll reach the day when all childhood cancer is preventable or curable. As always, keep up the fight, and thanks for listening to This Week in Pediatric Oncology.
We welcome your comments, questions, or thoughts on topics for future episodes. Just drop us a note at TWiPO@solvingkidscancer.org. You can follow Dr. Cripe on Twitter at KidsOncDoc, send an email to Dr. Weigel at weige007@umn.edu, and find all TWiPO episodes at solvingkidscancer.org.